Hi, I'm Jessica Fraser. I'm currently working on my master's at Université Laval in Quebec, but today I'd like to talk to you about research I worked on during and after my undergrad at the University of Victoria in British Columbia with Dr. Steve Perlman. I looked at whether carrying a selfish X chromosome could make Drosophila testacea larvae more likely to be killed by parasitoid wasps. Let's get right to it. So, a selfish genetic element is some piece of the genome that promotes its own transmission to the next generation, regardless of whether that's good or bad for the organism carrying it. Now, we don't actually know how common these are, since we can't always see them. They don't always produce an obvious phenotype, that is, they may act in ways that we can't see, and they can also be masked by suppressors. Basically, there's selection pressure on other parts of the genome to stop their own transmission from getting distorted by these selfish genetic elements, and they fight back. The image here is Barbara McClintock, who discovered transposable elements, which are one kind of selfish genetic element, but not the kind I'm talking about today. The type I am talking about today is sex chromosome meiotic drive, which is a case where the selfish genetic element is on a sex chromosome, and it acts during or immediately after meiosis to prevent the transmission of rival gametes that don't carry copies of it. Now, we don't know for sure exactly how common this is. Again, there are suppressors, for instance, that act in these systems. However, it is known from at least 15 species of Drosophila. It was actually first found in Drosophila obscura, pictured here. It's also found in a handful of other flies, rodents, and flowering plants. Now, I'm looking at a case in Drosophila where the selfish genetic element is on an X chromosome, and from this point on I'll be referring to it as a driving X chromosome, with the symbol XD. The D stands for driving, or you can think of it as distorter if you like. But basically how these driving X chromosomes work is that there is an element on the X chromosome that prevents a male fly that carries it from producing viable Y-bearing sperm. This ensures that any offspring the male fly has will have to inherit that driving X chromosome. The result is that this male fly produces only female offspring, and this sex ratio distortion is a phenotype that we can actually see and study. Now, something else that's interesting about these driving X chromosomes is that the selfish genetic element is generally found on a region of reduced recombination, because the genetic machinery needs to be in an area where it's not going to be broken up so it can actually keep on working. And in Drosophila, this generally means that the driving X machinery is found on a chromosomal inversion, which, roughly speaking, means an area where a piece of the chromosome got flipped around, so it still functions normally, but it doesn't recombine normally. And chromosomes with these inversions will end up diverging from chromosomes that don't have the same inversion. And mutations can potentially build up on the inverted region because they're not so easily purged through recombination. Now, the fact that these driving X chromosomes also promote themselves means that they might carry along some of these mutations with them, even if the mutations are somewhat harmful. And that's what this research really looks at, is whether these driving X chromosomes in Drosophila can carry harmful mutations that affect fly fitness separately from the sperm killing and sex ratio distortion. Now, there are some examples known of associated fitness impacts with driving X chromosomes. In Drosophila recens, females that were homozygous for a driving X chromosome were also found to be sterile. And in Certodiopsis dalmani, the wonderful stalk-eyed fly pictured here, males with a driving X chromosome were found to have a narrower eye span, and females prefer to mate with males that have a wider eye span. Now, these are still related to reproduction, but not necessarily directly related to the gamete killing of the driving X chromosome. Coming to my study system, previous research in the lab had pointed to some oddities in terms of the development of male flies carrying a driving X chromosome in the species Drosophila testacea. Drosophila testacea is a woodland fly widespread in Europe and Asia that feeds on mushrooms. It has a recently characterized driving X chromosome system, where the driving X chromosome was found to be significantly divergent from the wild-type X chromosome. I'm denoting the wild-type X here by XST. ST is just for standard. And there are suppressors of drive found in this species as well, but not in the lines of flies that I worked with. Now to those developmental oddities that I mentioned. This figure shows, from previous research, adult eclosion time, where day 1 is the first day an adult fly emerged, and then you see all the subsequent days. And these are for a group of Drosophila testacea males separated according to their X chromosome genotype. So the driving X males in orange here emerged later than the males that had a wild type X in the gray. And this phenomenon was actually not observed in females that were heterozygous for the driving X chromosome, which was presumed to be because they also have a wild type X to compensate for whatever was going on that driving X chromosome that was responsible for this. And the idea for the research that I worked on is that if driving X males are developing more slowly, that may be an indicator that they're carrying some kind of mutation that's affecting their development in some way, and that maybe the developing flies are also more vulnerable to stressors, such as the stressor I ended up looking at, parasitoid attack. So parasitoids. The term refers to a type of insect lifestyle where the insect lays its eggs in or on another insect, and then its offspring will consume the other insect, the host. So unlike a parasitoid that is successful in its attack always kills the host. So parasitoids do put quite a selection pressure on their hosts' populations. Now the particular parasitoid that I looked at, Leptopilina heterotoma, pictured here, is a common parasitoid of many species of Drosophila in Drosophila testacea's native range, and it very happily develops on Drosophila testacea. This particular parasitoid lays its eggs inside of Drosophila larvae and allows them to continue developing until pupation before killing them. Basically, the question underlying the experiment was, are male Drosophila testacea with a driving X chromosome more likely than wild-type males to be killed by these parasitoid wasps? And the broader question this was getting at is, can a driving X chromosome have additional fitness effects that alter the host organism's interactions with another species? So let's look a little at how the experiment was run. It started out with a controlled cross of Drosophila testacea, mating heterozygous driving X females with wild-type males. The result was eggs containing the four different genotypes that you see here, so a mix of wild-type and driving X flies. Although I was primarily interested in the males, because as I touched on already, they don't have a wild-type X chromosome to compensate for any effects the driving X might have. So I mated the parents for the cross I just described and allowed the female flies to lay eggs on discs of mushroom agar medium for 24 hours. Then I portioned those discs into groups of 50 eggs, which I transferred into vials containing mushroom and Drosophila food medium. Each vial was then randomly assigned to either the wasp treatment or the wasp-free control, and after allowing some time for the eggs to hatch and the larvae to start developing, I added 2 mated female Leptopilina heterotoma wasps into each of the wasp treatment vials for 2 hours to allow them to attack the larvae. I then allowed all of the insects in the vials to develop, monitoring daily for emergence of adult insects, and I collected all of the flies that emerged and genotyped the males for their X chromosome genotype. And then finally, when the wasps emerged in the wasp treatment, I collected those as well. So for the genotyping process, I used quantitative PCR to look at the dynein gene, which has a different melt temperature in the driving X chromosome than it has for the wild type X. The left peak in this figure corresponds to the driving X chromosome, and the peak on the right is for the wild type X chromosome. So the main idea here was to compare the proportion of males carrying the driving X chromosome in each treatment, with the principle being that if the X chromosome genotype had no influence on susceptibility to wasp attack, then the proportions of driving X males emerging in each treatment should be identical, even if the overall number of flies was lower in the wasp treatment. I analyzed that using a generalized linear model with a binomial distribution, looking at the proportion of males with the driving X chromosome within each vial to see if that was affected by the wasp and control treatments. And then I also looked at whether the male emergence time did end up depending on the X chromosome genotype. For that, I took the log-transformed mean emergence time for each genotype by vial, and analyzed that with a one-way analysis of variance. So let's look at the results. Here you can see the overall emergence of flies and wasps in each of my experimental vials, with the wasps in red, and the wasp treatment there on the right. So there are a few things that are important here. First off, the rate of successful parasitism in the wasp treatment was variable but never overwhelmingly high. That's good, that was the goal, so that there would still be enough flies making it to adulthood to yield a sample size that we could actually work with. Also, we started out with 50 fly eggs per vial, and the emergence here doesn't really get above 40, with some vials being quite a bit lower than that. Emergence overall was quite variable, and the overall emergence in the wasp treatment was a little bit lower generally than in the control. Finally, if you look at that x-axis, you might notice the numbers. There's a couple numbers that are skipped. That's because a couple of vials were actually omitted from the genotyping and analysis on the basis that vials with extremely low pupation rates probably had something severely wrong with the conditions in the vials. But coming now to the results of the male X chromosome genotyping, we've got the flies broken down here by genotype. Driving X is in red and wild type is in the dark gray. And you can see that the control has a slightly higher proportion of driving X males than the wasp-exposed treatment. However, again, there was quite a lot of variability. It's a little more obvious, the trend with the box plot here. You can see there's sort of approaching 3/4 driving X males in the control and closer to 50-50 with the driving X males in the wild type in the wasp treatment. But statistically speaking, with that binomial GLM I mentioned, the difference was not significant, with the p-value sitting around 0.09. Finally, looking at the development times in the males by genotype, the difference that was previously observed between the male wild type and driving X emergence times didn't show up here. You can see above the emergence time by genotype in the control group males and below the wasp treatment males. And in both cases, there's really no difference between the two genotypes. Interestingly, you can see the control group had a few more stragglers, but those were actually of both genotypes. And again, you can see the trend very clearly in the box plot. There's really not much of a difference at all. Statistically speaking, that ANOVA showed a p-value sitting around 0.48. Definitely not significant. So what do we draw from all of this? We did notice that trend showing a lower proportion of driving X chromosomes in male flies in the wasp treatment compared to the control, although it was not statistically significant. There were also a few other interesting things that showed up. Contrary to earlier findings, we didn't see this time that slower development time in the driving X males. And there was also that very high proportion of males carrying the driving X chromosome, particularly in the wasp-free control. So if we consider that sort of lower but non-significantly different proportion of driving X males in the wasp treatment, we might wonder what happened there. Well, it is possible that the driving X chromosome doesn't affect Drosophila testacea's defenses, or just doesn't affect them against this particular natural enemy out of all the natural enemies in the world that we could have picked. There was some recent research that looked at whether driving X chromosomes in Drosophila affinis and Drosophila neotestacea affected their carriers' susceptibility to two bacterial pathogens, as well as they also looked at an autosomal distorter in Drosophila melanogaster. Now, the results of that research were quite mixed, depending on the specific driving chromosome and on the sex of the fly. There were some instances where the drivers seemed to have an effect, making the flies either more or less susceptible, and it was sometimes statistically significant, sometimes not. Overall, this sort of leads me to think that this is just a very complicated question to answer, and very context-dependent. Looking at this experiment, between the variability observed in terms of emergence and the sort of presence of confounding factors, I don't know that I would treat this as a sort of conclusive negative result, maybe more of a jumping off point for further research. So as I've already indicated, there was very high mortality overall. I did start with those 50 eggs per vial and got sort of much lower emergence than that in many cases, suggesting that perhaps the conditions in the vials weren't optimal for the flies and they were dying young as a result of that, which is a pretty big confounding factor in an experiment on causes of fly mortality. There may have been some cases where the parasitoids attacked larvae but then failed to complete their own development, which would be quite interesting and might explain the lower emergence in the wasp treatment. Future research might get a little bit more clarity by looking at the larval stages as opposed to the adult stage, although there are logistical challenges to doing that. And finally, there was a sort of a relatively low sample size, 12 vials analyzed per treatment, for detecting differences given the amount of variability we ultimately did observe. But something very interesting happened in the control in particular. Based on Mendelian inheritance, we would expect about 50% of the males to have inherited a wild-type X chromosome and 50% to inherit a driving X. And in the absence of factors favoring one type of X chromosome over the other, we would expect that ratio to have been what we got in the adult flies, 50-50. That's not quite what we did get. The proportion in the control, as you might recall, was closer to 3/4 driving X. Now this actually lines up with some other research that's still ongoing in the lab, looking at whether these particular driving X chromosomes might actually be able to distort transmission in females as well as in males. Now that would be highly unusual. I don't actually know of any other systems where it's been demonstrated, but if that's what's happening, then I mean that's very interesting. But also it would mean that analysis of this kind of system becomes even more complicated because you can't compare the proportion of driving X males in the wasp treatment to the sort of expected 50-50 baseline, but rather to some unknown, possibly variable baseline transmission of that driving X chromosome. And then looking at those differences or absence thereof in terms of male development time between the X chromosome genotypes, I think I know why we got contradictory results. The development times in the previous research were looking at eggs that were laid over several days in a row, and then the emergence was checked every day, whereas in my research, the eggs were laid all within the same 24 hours, and then the emergence was checked every day. So in the earlier trial, it's possible that those emergence differences didn't come from larvae that hatched at the same time developing more slowly, but rather something like differences in how the eggs were actually distributed. And I think it would be quite worth looking into, seeing how those different factors might have affected it. Ultimately, I think this is something that's worth continuing to look at. Hopefully future research can further build on this work and maybe avoid some of the pitfalls that came up. I think it would be very much worth looking at whether mutation-induced fitness effects on these chromosomes can happen in female flies. That is something that I started, but there were some technical difficulties. It's not finished yet. I think whether or not future research ends up turning up significant effects of the driving X chromosome, specifically on susceptibility to this parasitoid, it would be worth examining other areas where it might potentially have effects, for instance, larval competition. And then if this trend of lower proportions of driving X males sort of emerging in the wasp treatment actually was, if it was confirmed by future research, if it turned out to be significant, that would open up the door to another big question, which is what is actually the mechanism for that? It could be related to a greater probability of one genotype being parasitized to start with, for instance, differences in larval behavior, or perhaps to a greater probability of the larvae dying after being attacked due to differences in larval immune responses. So overall, I think this is really a fascinating field for future research because the question of how genetic parasites like this selfish X chromosome might affect ecological interactions, including what we think of as a more conventional kind of parasite, this parasitoid with the fly, it's still quite an open field, and I look forward to seeing what develops. I would like to thank my wonderful colleagues in the Perlman lab for all of their help and support with this project, as well as my sources of funding. And thanks to you, my lovely virtual audience. If you have any questions, please reach out to me.